Hello everyone, welcome to today's video on A-level biology revision. For anyone who might be their first time here, I'm going to be teaching, talking through basically, because I'm not a great teacher, about a little subtopic in biology, just to help me get my head around it and sort of understand it with you guys as I'm learning. I'm not sure if I'll post episode 1, a little bit of shambles, haven't even finished what I needed to go through in that episode, so here we are. Today, just going to do a quick one because my voice is kind of going, and it is kind of late at night. Not as late as night as the first one, but so today I wanted to go over biological tests for molecules. So I should have brought my coloured pens, but I didn't. So that was silly. So biological tests for molecules. There's 5 you need to know. You need to know test for sugars, reducing and non-reducing. That's done by the Benedict's test. You need to know the test for glucose, test for iodine, the iodine test for starch. You need to know the test for protein, which is a Burette test, and the test for lipids as well. I think that's 5. Starch, protein, lipids, glucose, and sugars. Yeah, so let's just write this down for you guys. So we're going to start with Benedict's test for sugars, and so there's 2 types of sugars. There's reducing and non-reducing. Reducing monosaccharides, all of the monosaccharides, and some of the disaccharides like maltose and I think sucrose. I could be wrong, but no, it's lactose. I take it back, it's lactose. And this is sort of the basic test that you do for you'll see what I mean when I talk about non-reducing sugars. But for what you do for the Benedict's test for sugars, you add Benedict's reagent, kind of makes sense, which is a dark blue colour, like this pen. It's kind of a dark blue like that colour, and you heat it in a water bath that has been brought to boil for, I don't know, 5 minutes maybe, and a positive test result will be a brick red colour, although it's on sort of a colour scale from dark blue, which is where it begins, to brick red, so you can get like green, yellow, orange before you get to brick red. And the colour, the intensity of that colour, like if it's red, indicates the concentration of the reducing sugar. So if you've got a brick red solution, it's going to be more concentrated than if you have an orange or a green colour sample. Obviously, if it stays blue, then there's no reducing sugars present within this solution that you're testing for, and you can rule it off. But a more accurate way of doing it is to work out like the concentration of something or the mass that's in there, is to filter the solution the way they precipitate, but that's kind of long, so I wouldn't personally recommend doing that. I'm just going to write this down. So Benedict's test, warm in a water bath, blue, if I have my colours, would be so good. Blue to brick red. Yeah, so that's for reducing sugars. For non-reducing sugars, it's a bit different. You first got to break down the non-reducing sugar into reducing sugars through the addition of dilute hydrochloric acid. This just breaks the bonds between, I don't know, whatever it is, the disaccharides, polysaccharides, and breaks them down into the monosaccharides so they can be tested. So you basically just get your new sample, you add a little bit of dilute hydrochloric acid and heat it in the water bath. You then need to add a little bit of sodium hydrogen carbonate before you can put it back in, and you just leave it in there in the water bath, and it will be the same thing as a reducing sugar. It will turn from blue to whichever colour is in the spectrum, whatever precipitate it is. Obviously brick red, most positive, most concentrated. Blue, no sugars present, reducing or non-reducing, if that's the case. Yeah, I think that's something you need to know for the Benedict's test. So quickly recap, positive result is from blue to brick red along like a colour gradient. You warm in a water bath with Benedict's solution, Benedict's reagent, sorry. And yeah, you can weigh the precipitate, filter out, weigh it to work out the actual mass of it. But for non-reducing sugars, you've got to add dilute hydrochloric acid first, put it in the water bath, let it warm up, then take it out, add sodium hydrogen carbonate, and then pop it back in, and then add the Benedict's test and leave it in there, and that will give you your result for the reducing sugar, non-reducing sugar. That's the whole of Benedict's test for sugars. Then you've got glucose. How do you test for glucose? Well, there's two main ways. A sort of like standardized way that's not quantitative. You can use test strips, which are useful for diabetes patients to test if there's glucose present in their urine, because obviously glucose shouldn't really be present in urine, so that's when they know, oh no, I need to get something sorted. The other way is to use colorimetry, and you can use Benedict's reagent and add it to a colorimeter, and a colorimeter basically measures the absorption of light through it. So when you use a colorimeter, you're supposed to calibrate it first. So you use water, which has a 0% absorption, 100% transmittance, and obviously the color that your solution will have different levels of absorption, which link to a concentration, and it basically just tells you how much glucose there is. So if the higher the glucose concentration, the lower the absorption will be. That's just the general rule for glucose and colorimetry. So the test for starch is the iodine test. Very simple. It's just iodine dissolved in potassium iodide solution. Do this test sample, just add it in there, and it will change from browny orange to a blue black in colour. So like, don't really have anything that's like sort of like browny orange up there, browny orange, like kind of like my wall. My wall's kind of, no, I'm capping. It's not browny orange, but just to a dark bluey black. It's very obvious, very quick change, and that will just tell you if starch present. If there's no starch present, stays orange and black or whatever. No, it doesn't say black. Stays orange. Stays browny orange. I lie. For the burette test for proteins, very simple as well. 2 stages. So it's important to note that the test must always be alkaline. So you first add a few drops of sodium hydroxide solution, and then you add a little bit of copper 2 sulfate, and that makes your burette sort of like reagent sort of thing, which is in there. And if the protein is present, it turns purple. If not, it stays like a bluey colour. So a good way to remember this is, if you remember that it goes from blue to purple, if you think about blue, what do you know is normally blue? And at least for me, my thought is to go straight to copper 2. That like jogs your memory. Oh, copper's blue, so what's added first? Copper 2 sulfate. Then you just got to kind of remember that it's got to be alkaline, and sodium hydroxide solution is supposed to be added. You've got to just kind of remember that. It's one of those things. So yeah, blue to purple. Obviously, if there's no protein, stays blue. Yeah, and then finally we've got the emulsion test. Emulsion test, very simple. It's you add ethanol into the test tube, and then you shake it for about a minute, and then you pour that solution into water. If a lipid is present, the solution will turn like a milky colour. If not, then obviously there will be no precipitate, like no white milky precipitate. The more lipid there is, the stronger the milkiness. Not sure if that's correct phrasing. The more noticeable the milky colour will be, basically. And yeah, so just a quick recap of everything we just said, just so you remember. Right, so we've got the Benedict's test. Right, I'm always got my whiteboard up, but I never write anything. Anyways, so that's the point. Benedict's test. That is a test for sugars. You've got reducing, non-reducing sugars. The same colour gradient for both of them. You've got from blue to brick red, and it's colour gradient, so it goes like blue, green, yellow, orange, then brick red. And you warm in a water bath that's been brought to a boil, and for reducing sugars at least, that's what you do first. For non-reducing sugars, you've got to first break down the non-reducing sugars into reducing sugars by adding dilute hydrochloric acid, chucking it in the water bath, taking it out, neutralizing the dilute hydrochloric acid with sodium hydrogen carbonate, sucking it back in with the Benedict solution, leave it in there. Then you look at your colour scale, if it's gone brick red or if it stays blue, and that tells you how much solution is present. You can also use colorimetry in here as well to see the absorption of the colour to work out the concentration in there. You can also filter and weigh the mass of the precipitate, and that also gives you an indication of how much was in there. Then the iodine test for starch, that turns from browny orange to dark blue in colour, and it's just iodine dissolved in potassium iodide solution. Obviously no starch, stays brown. Purette test for proteins, bluey colour to a purple, so pale blue really. And first you've got to remember that the solution's always got to be an alkaline, so to do that you add sodium hydroxide, which is an alkaline base, and then you've got to add copper sulfate solution, because it's got to be blue beforehand, and copper 2, as we know, is blue, so it goes from blue to purple. Purple, positive test. Obviously no proteins present, stays blue. As for the emulsion test, you add ethanol, you shake it for a minute, then you add it into water, and if a milky white precipitate occurs, then you've got lipids present. If not, no lipids present. The more milky and white the precipitate is, the more lipid is present in it. Which one have I missed? Oh, test for glucose, test strips, and then colorimetry. And you just got to remember, the more concentrated the colour of the glucose from the Benedict test, because you're adding the Benedict test for the glucose, because it's glucose is a sugar. The more concentrated the colour, the higher the absorption would be. Just got to remember to always calibrate your colorimeter before you use it, which is you use that using water. That's quite important. And yeah, that's all of everything you need to know about biological tests for molecules. So good luck with my A levels and yours.